I welcome you all on this eFDP program and in the technical session 2, where today we will listen our chief guest, our speaker of the day, Professor S.S. Gaurav. He is Dean, Faculty of Agriculture, CCS University Meerut, and he will be speaking on use of nanotechnology in disease and pest management. So he will be joining us very soon. In a couple of minutes, we'll start this program, today's technical session.
Not open.
So very good morning to all of you. I'm pleased to welcome each and every one of you. Today we are going to start our technical session 2, in which we will have a very important discussion on the topic of use of nanotechnology in disease and pest management. Today we have got our eminent speaker, Professor Singh Gauravji, Professor S.S. Gauravji. He's the Dean of Faculty of Agriculture in Charan Singh University, Meerut, and he'll be speaking on this topic. So, dear friends, I would like to mention our chairman, Dr. Virendra Singh ji here, co-chairman of this faculty program, Dr. Ramesh Pal, and Dr. K.P. Chauhan ji, organizing secretary, Dr. Satyavan Singh sir. So let's start today's section without delaying any much of time. So today's speaker, as I told you, he is Dr. S.S. Gaurav. He is a learned professor, genius scientist, excellent administrator, and moreover, a very humble and down-to-earth human being. And we have got many interactions, academic and other than that. So today, really, we are fortunate enough, sir, to have you and to start a technical session with you. I would like to mention about the Dr. Singh Gaurav. He has got his doctorate degree from the Pioneer Institute of Agriculture Research in India, from Indian Agriculture Research Institute, IRI, Pusa, New Delhi. And presently, he is working as the Dean of Faculty of Agriculture and Professor and Head of the Department of Biotechnology, Seed Science and Technology. He is also holding the position of Nodal Officer and Coordinator of Intellectual Property Cell, All India Survey on Higher Education Cell. And presently, he is engaged in many kind of research, education, administrative, and extension activities. In research activities, he is working on the different area of plant breeding, seed technology, plant biotechnology, to address the local concern of Western UP that includes morphological, biochemical, and molecular characterization and evaluation of genetic diversity, heterosexual breeding in various crops like wheat, rice, lemongrass, basil, chickpea, peas, etc. Recently, Professor Gaurav and his team have initiated their research work to exploit the possibilities for the development of nano-biopesticides against early and late blight of potato and white grub of sugar cane crop. Dr. Gaurav is also engaged in teaching of master's, MSc and MPhil and doctorate programs. Besides this, he has supervised 15 PhD students and 25 MPhil students and more than a dozen of MSc students in the area of seed science and technology, genetics, plant breeding, and biotechnology. He has an excellent academic career, having won gold medal for his MSc AG degree from MPKV Rauri, Maharashtra, and he has been recipient of more than 15 awards from different scientific bodies in India and abroad. He is awarded by the Best Teacher Award by the Honorable HE later Sri Ram Naresh Yadavji, Government of Madhya Pradesh, and he's also several other awards like Scientist Leader Award from IIRR Hyderabad in 2015, Science Initiator Award in 2016 by Rajmata Sindhia Agriculture University, Gwalior, Madhya Pradesh. He is known for his event organizing skill, and he has organized more than 10 national and international conferences, which out of which 4 conferences were held in the outside of India, and with the collaboration of the foreign university. like Kasarset University, Bangkok, Thailand, Nanyang Technological University, Singapore, Nong Lam University, Ho Chi Minh City, Vietnam, and Masa University, Kuala Lumpur, Malaysia. And I'm a proud member of the organizing committee, and I can say with proud, with privilege, that I have been a part of these assignments, these conference with you, sir. Dr. Gaurav has visited many countries of world for the professional cause, for his academic purpose. Most of the, like France, he visited France, in the National University of Kyiv, Ukraine, in Malaysia, Singapore, and Thailand. He has got published about more than 102 original research papers in national and international repute, and one patent credited to him. He has also authored 6 textbooks, 10 popular articles, 4 book chapters, and more than 100 abstracts. He's also the author of 6 NAS rating, he's editor of 6 NAS rating journals, and handled 3 major research projects. There is much more to mention about Sir. He, on the basis of his credential, he is just nominated by the President of India as a member of Executive Council of Allahabad Central University, Prayagraj, Uttar Pradesh. And there is much more remain to mention about him. And we people who has spent a little bit time with him are the testimony of his bright career and his potential. So without taking much time, very humbly, I would like to request to Professor S.S. Gaurav to kindly take the mic and take this virtual platform and start your presentation, your views on the use of nanotechnology in disease and pest management. Over to you, sir.
Thank you, Dr. Himanshu. I am audible to all?
Yes, sir.
So really, it's very big and exhaustive introduction about me. So first of all, I would like to convey my regards and congratulations to all the team members. Those are organizing this very specific 1-week faculty development program on inclusion of innovative research in education for smart agriculture that is started from 7th and that will be ending on 13th of May. So I humbly congratulate to Professor P.M.B. Pandey, who is the Vice Chancellor of this university, IFTMU, co-patron Professor Sanjeev Agrawal, and no doubt the more important, the chairman of any conference, Dr. Birendra Singh. I visited IFTM and I met him. So I remember that my memories with him. And co-chairman, Dr. Ramesh Pal, Dr. Kishan Pal, and no doubt convener, who is currently introducing myself and very active and very energetic and intelligent person, Dr. Himanshu Trivedi, and organizing secretary, Satyabhan Singh, co-organizing my very good student and now my colleague, I can say, Dr. Spal Singh, who is always sincerely devoted during his stay in PhD program in this university. And in every and each when I remember him, he always be present for any social and academic cause, as well as other coordinators, A.N. Chobe, Sanket Samant, Abhay Saini, and I can also see Sudhir Kumar Dhamaji in his video. He is also present. He is one of my friends. So really, first, before starting, I am very happy that I have been invited and given this opportunity to interact with you, and I congratulate the whole team and all the participants. So, my slides are visible to all?
Yes, sir, it is visible.
Okay, thank you. So, as I have been assigned to talk about use of nanotechnology in disease and pest management. So, with this topic, I'm going to talk in detail. And first of all, I will try to talk about the major content. That whole topic is divided as an introduction. and applications in agriculture and even in medicine also, application in medical sciences and significance. And we will also talk about what we are doing in our lab. So first, I think basically I prepared this slide in two format. One that when students, those are connected, though the mass of students, whether other than trainees, if any students for PG or UG, they can also be benefited, as well as definitely those are all on the participants. So nano, we all know that it is indicate the dwarf, but how much dwarf? That is about 1 millionth part of a meter. So when we talk about a nano scale, so it ranges about zero point 0.1 nanometer to 100 nanometer in range. So nanotechnology is nothing but it is like other sciences. This is also a specific science that manipulating the matter at nanoscale. When we uh, go for the working of any item in the form of nanoscale, that in already in that above range, then we that may be in the form of design, in the form of characterization, production, synthesis, application, structural, or controlling the shape and size of any article or finished article or any biological item that comes under this category of nanotechnology. To understand that how smaller nanoparticle is, this is the blood set about 2 to 5 nanometer. If we go for the width of a double stranded DNA, about 2 nanometer. So this is the biometer, there's a 10 nanometer, nanocells, 2 to 20 meter. So this is the simple example by which we can imagine that when we talk about 1 nanometer, how smaller it is. When we talk about nanotechnology, this is a very old age technology or science. If we could remember when old days or when our grandmother or our mother crosses through any river or any pond, those are of any ethical nature or holy nature. Normally we used to place, earlier the coins were of gold or the silver. So we used to dash coins to the water by saying that we were used to worshipping by putting the coins. But that practice was basically uh, maintained only to create the nanoparticles of this gold or any other this compound. That the synthesis was after a year and a year, the synthesis of particle, they given the basically antibacterial, anti-fungus characteristics that was making pure or purification of water. That was the old age practices. But earlier time, nobody was aware why they are doing. That was just was simply the practice of the worshipping of a river, of any water bodies. So if we could talk about this technology was also very old days, about 400 AD, that there was a, in case of Damascus and Romans, they have designed a cup that was the gold silver alloyed nanoparticles. And in this cup, this was basically when this light was reflected, then it was reflecting green. When light was passes through this cup, then we can see how the color was changing from this green to the brilliant red. So this was due to the gold silver alloyed nanoparticles. This is very old. They have discovered this technology. Similarly, another example we can see about 900 CE, Maya blue. The paintings were found. Still, they are present in Mexico. That even after thousands of years, still the paintings are uh, quite good in condition. Only due to that, they have been basically Maya blue color was basically designed and painted by maintaining the nanoparticle size. These are the example of such old age practices. This is the another example that the sword was they were maintained in near east, and they were basically imported from the southern India. So this is the technology also available in southern India. So this is about 300 to 1700 AD. So we can see these short, the flower-like structure, many kind, this ladder type of structure, they were maintained by the some compounds at the nano scale, and that this such kind of by the using this was capable of being very sharp and resilient edges. So similarly, these another example of luster decorative metallic plate. These are the simple plate, but they were created by the controlling copper and silver compound at a very low temperature, about minus 650 degrees Celsius. So they were so strength was so that they were not disturbed by any temperature or by any kind of humidity or by any climatic condition. So these are the creation of about uh, 10th century 10th century of silver nanoparticles. So we go to the historical management. Nanoporous ceramic filter was indeed already being used 19th century, that to separate viruses. In 1902, 4 nanometers were successfully in ruby glasses microscope they were developed, and this one of the Nobel laureate Richard Feynman, that one of the physicists, he has already won Nobel Prize by using a different kind of use of nanoparticle. So this is the basically historical aspect from here. The advance of the nanoparticle were started from 2000 year ago. So now, since that year, now we can say that about in this now nanotechnology initiative group has been launched in the different countries. In large number of nanotechnological missions are utilized. Please, sorry for interruption. बताइए अभी नहीं बंद करो sorry. So now each and every country there are nanotechnological missions or several other agencies. Those are maintaining different kind of research in all the areas of nanoscience. These are the different kind of compounds: gold nanoparticles, polymeric, dendritic, micro nano. So functional carbon nanotube, different kind of compounds by which nanoparticles can be created with the different size, then different size shape. To understand what is the basically strength of nanoscale, if we see the normal this graphite pencil, when we are using this graphite only in the form of pencil, this is very weak. We know very soft, we can break very easily. But when we this press this graphite at the nano scale, or we can make this nano scale, then the same graphite will be stronger than the steel and 6 times lighter. So this is the basically importance of this nanotechnology. Similarly, if we go to the copper, this common copper, if we go for the nanoscale, these copper metal, they may be not only there will be more strength, about 50 times of strength without any breaking. So 50 times strength increasing only if we do the same, this elasticity will increase 50 times. And similarly, these are the orange gold. Uh, if we go for the nano scale, then different kind of color can be acquired on the basis of size and shape of the these orange, yellow, gold. So these are the different simple example to understand the students those are interested to do in nano science. There are basically for the purpose of production of nanoparticles two major approaches. In one approach, we call it top down. In top down, basically this is the macroscopic, about very bigger size can be broken into the very smaller size. Means about 10,000 nanometer size. If any compound or any article of the 10,000 nanometer, then by this approach we can go for the up to the this maintaining of 1 nanometer. And another is the just reverse, that we call is bottom up.
In bottom up, a smaller particle can be arranged to utilize in a macro form, like gel precipitation and other processes. So these are the different uses of nanomaterials. Is the medicine we can use for the cancer treatment. There are already this is under use in cancer treatment, bone treatment, drug delivery, appetite control, drug development, different kind of medical tool, diagnostic test, imaging. So other than this, other several all kind of medical activities now nanotechnology is the one of the this common tool, but it is only the advanced tool, common but advanced tool that is used by medical science. In case of energy science, efficient this is used for the solar cells, fuel cells, and batteries. And food and beverages, now in market, large number of beverages are available. Similarly, in case of information technology, uh, this, this is going on, and for other kind of, so each and every area now nanotechnology has made its encroachment. In case of agriculture, basically we use it for resource management, to minimize the basically management of the waste production, and agricultural nanosensors and other. Now if I think you all have read in the newspaper that nano urea that by delivered by IFCO already came into the market. Earlier also some fertilizer and pesticide are available, but public sector, this is the first time that by the government of India authentically nano urea has been given. And priming agents, we have also worked in the area of priming by the nano tubes. So these are the another different applications in all the agriculture field. So we can say each and every agriculture field is where we can use nanotechnology very easily. Even as I am the person of genetics and plant breeding, so putting this example is also important. So for the production of transgenics, we can go for directly uh, insertion of DNA into the cells with the help of 3 nanometer mesopore silica nanoparticles. So go for also DNA sequencing, we can use it. And in case of seed technology, for the germination and priming, the one of the major important in increasing germination viability as well as the priming, we have already used it and very successfully, and by the it has given good results. The another way to understand nanoscience, they are now with the help of nanoscale level, this any self-healing material can be basically prepared with the help of these material, especially a packaging material. So contamination of microbial can be avoided if this polythenes or polybag or any coating material that is for the food and agriculture or any other item, if they are maintained, if they are made with the uh, using nano scale level, then that may be work as a self-healing material, means they will not allow to germinate any kind of microbial contamination. So there is no need of another any other kind of use of any way, so to maintain or such kind of material. And similarly, we can design the sensor with the help of nanotechnology, and that sensors are having so much of strength that the thing that our nose cannot detect, this sensor can detect. So before going for any spoilage, we can check it or such kind of spoilage, and we can initiate our control damage. Similarly, very soon the work is going on that in very near future we can get some pesticide and herbicides with a very lower doses. I think within 5 or 10 years, rather than normal chemical pesticide, they will be replaced with the weedicides or herbicide or other pesticide by the nanostructure material. Now I'm shifting to my work. So our work that has been completed by two, this is the compilation of my two students. One of the now PhD has been awarded. One is Dr. Amardeep and another is Dr. Janika Sukla. So these we have taken these major four objectives, that biogenic synthesis and characterization of silver and zinc nanoparticles. And our second objective was comparative analysis of nano biopesticide with commercial chemical pesticide. And third objective was evaluation of toxicity of nanopesticide to plants and environment, and liquid formulation of NP-based nanopesticide. As we are going for the patent of liquid formulation, so I will not disclose, I will not present about this. We have applied for the patent, and very soon we are expecting to get it patented. Then we will able to present it in the public forum. So in biogenic synthesis, for this purpose, we have utilized Aspergillus niger, a fungus, and the fungal biomass was prepared, and that fungal biomass, the extraction of fungal about 50 ml of fungal biomass and 950 ml of either silver nitrate chemical or zinc nitrate. If we are going for the this synthesis of zinc nanoparticle, then we have to use zinc nitrate. If we are going to synthesize silver nanoparticle, then we have to use silver nitrate. So this solution was 2 millimolar. So by about 72 hours to 120 hours, incubation was done. But our results, the firm results, those we have basically utilized, they were of about incubation of 120 hours. Then we go for the photometry. So on this, the color changes. This is the basic Aspergillus niger. This dark dark to this light kind of color was obtained when synthesis started. So we can see that after 120 hours, this is of silver, and this is about brown color was obtained after synthesis, and pale yellow color was in case of zinc nanoparticle. And similarly, another approach was the plants. So earlier synthesis was done by the fungal, using fungal biomass, but similar kind of approach was done when we, in place of fungal biomass, we utilized bamboo leaves or tulsi leaves. That in place of one gram of fungal biomass, we have replaced this extract of bamboo leaf of this leaf, and after boiling of 30 minutes, we obtain, and then again incubation was 72 to 120 hours. So the other procedure is similar. So this synthesis was this quite light brown color or brownish yellow color was obtained when this was synthesized by using the nanoparticle of this silver by the tulsi plant, and this is the more darker when we have used Bambusa vulgaris, that is bamboo leaves. This is the standard range for the spectrophotometry, and we have utilized different kind of techniques to characterize and to know the shape and size and quantity of nanoparticles. So this is the normal indigenous method to examine whether the solution after 96 hours the nanoparticle has been synthesized or not. When we place normal laser light, if laser light is passes through without any scattering, it means there is no synthesis. But if the laser light is scattered, as we can see that is scattered, and it does not passes or passes very slowly, and whole latter is scattered, it means there are presence of nanoparticle has been synthesized. This is the normal and indigenous method that is used to know whether the nanoparticles are synthesized in our solution. This is the simple preparation for SEM and ICPM. So with the help of spectrophotometry, those uh, they have been prepared by using Aspergillus niger silver nanoparticle and after 120 hours. So when we go for the 320 and the peak were obtained at the 340 and 430 and up to the 460. So this confirms that presence of the silver nanoparticles after 120 hours of incubation. Similarly, in case of this JDN, the peak were obtained in 235 and 275. That will it shows that presence of zinc nanoparticles. And this is similar kind of study was in case of when the plant extract tulsi was used. Here the peak was obtained at 405 to 475 nanometer. So at this wavelength, this presence of these nanoparticles was shown. So these are the conformity study, and then there is a similarly for the tulsi plant for zinc particle. And for to know, the scanning electron microscope was used, and for this purpose, we have outsourced from IIT Kanpur, and this is to know the what are the different size those are obtained, and these are the basically different size. The range of the size we obtain 20 to 50 nanometer in our synthesized Ag and EP. This is the range of zinc nanoparticle by using the same method. And in case of basically energy dispersive X-ray, that is also called in short form EDS. By using EDS, this is known that other than our compound nanoparticle, what kind of different compounds are normally found in the solution. So as this aluminum foil are utilized for wrapping of the material, so you can see that aluminum presence of aluminum was also shown other than Ag. And similarly, in case of zinc, we have also see the potassium, and for this purpose, then several filtration are normally done to make it purify. So by this method, we can know that what kind of other compounds are present in solution, and that can be just removed by the filtering method. These are the another approach, dynamic length scattering. This we have utilized from the Motilal Nehru Institute of Technology from Allahabad. Here we can see the range from about 8.9 to this about 227 nanometer, and this goes up to the more than 68 of range. So this is the for the distribution, size distribution of range. So these are the different techniques and equipment those are utilized to characterize and to know the distribution of size. So after synthesis and characterization of these nanoparticles, then we have utilized different different kind of solution with different ppm and molar to our crop. So for this purpose, we have taken basically three. One is Fusarium oxysporum, Alternaria solani, and Phytophthora infestan in field. And we created one field. We created natural, we allow natural growth of the disease, and in the same time we also this spread disease spore to create artificial the creation of disease. So by this, and another approach was that a white grub, it is one of the insect that is highly found in the sugarcane belt, and this is the 4-star insect, egg stage, larva, pupa, and adult. And this is very highly, larva is highly damaging, that cut the root of the, those are the from western UP land or sugarcane belt. They understand the problems those are connected with the white grub larva. So with this larva, this is very damaging, and adult is also sucking the sap of the leaves. So even for the white grub control, government of UP, they have initiated several kind of task force. So we have also tried to control the one. We have taken one insect and three fungus. So for we have applied all for these. So in case of first in fusarium, in case of fusarium, in vitro condition, we have basically applied this in natural growth when this is Photophthora and the culture of fusarium. So you can see that this was inhibition zone was reduced about 70%. If we quantify, this is the zinc, these are the AgNEP, and this was about 1.989 diameter, and the centimeter of height, this was the 0.093. So it reduced about 80-90%. So this was comparatively very good to control this Phytophthora. A similar kind of results were also obtained when the whole, in case of control, the spread of this colony was about 2.78 centimeter diameter, while here it was only 1.73. So about 60% this control was found. This is the field where we have grown our potato to treat. These are the different chemical and compare these twin 80 and twin 80 in 2000 ppm, 1000 ppm. These are the different concentration of silver nanoparticle with the aspergillus niger, 100 mg biomass, 1 mg biomass, 100 mg biomass, 2 molar solution, 1 molar solution. Similarly, the aspergillus flavus fungus, and this is the for this control. So different fungus were utilized and compare was done. And then these are the pea plant as fusarium. For the purpose of fusarium we have utilized. And this is for insects, and we have taken basically Meerut and Muzaffarnagar area, and about randomly the field was selected of those where the sugarcane crop was standing. 18 to 18 to 10 inch this pit was dug, and this method is called basically compact method, that was given by Lobe et al. In 2011. And when the randomly this was surveyed, that it was found in case of Meerut, average of 0.16 white grub in per pit was found, and similarly that was the above the threshold level, 100% effective, both zinc nanoparticle as well as silver nanoparticles. So this is the experiment conducted with the silver nanoparticle. This is the full fledged larvae. When the treatment was given of silver nanoparticle, then we can see the larvae was started shrinking, then more shrinking, then it was dead, and after 48 hours fungal growth was started after death. So this very effectively results were obtained in both the cases, zinc nanoparticle and silver nanoparticle. We have also compared the results of metarhizium and Beveria viciana, because this is the two items, those are very easily available in market to control these insects. So when we compared, then we found even after 10 days, maybe it depends to company, so we are not making any specific comment, but what we acquired in our results, then we got after the treatment of both these two. This is the treatment given of the metarhizium inisopile, and this is Beveria viciana. In both the cases, even after 10 days of incubation of treatment, the larvae were not dead, and no fungal growth was obtained. You can see that there was live larvae was obtained. So the result was not satisfactory, and we compare these two products, especially from here we obtain, they were not good for the control or management of white larvae. And similarly, a lethal dose were required for tomato plants also we have conducted. So by in conclusion, already we have this prepared the liquid solution of silver nanoparticles as well as zinc nanoparticles. In both the cases, very effectively, we are going to apply for the patent, and already patent is under process of the drafting. And I am happy to share that as the result, as I have talked to the patent attorney, they are very sure that we'll get our patent, and if the things will go as per our thought, then very soon we will come with this liquid formulation to use at commercial level and very cheap product to the farmers that will be available. So in conclusion, I can say that nanotechnology provides comprehensive insights, including relevant information on environment, impact, and safety, and have the role of potential application nanomaterial in crop protection and green and clean agriculture. It presents a strategic, comprehensive, and forward-looking approaches. So this is all about my talk, and already I have given my the research work. These are the some publications, and more publications have. This is a old slides about 6 publications with the international repute. They have been published, and this is in the local media, news in local media, and this is our lab. So these are my, I have got the 2 students, one from Vietnam, PhD student, one from Thailand. They also came in our lab for the training purpose, and his work, the whole work is of Dr. Poonam Rani, who is from Banasthali. She has also came here to complete her PhD program. Amardeep, who has already got his PhD, and Ganika, and these are the some administration. Those were basically connected with our lab during this work. So with all this, I acknowledge my former Vice Chancellor, who has provided FTIR and AAS about 1 crore instruments to my lab, and other funds support from IGIB and other organizations. So thanks to all for patience hearing.
Thank you very much, sir. Indeed, this lecture was fully it is what saturated with the informations, and it was blended with the findings of research work carried out by you and your team. Definitely, sir, nanotechnology is generally considered as somewhat complicated subject, but the way you presented it, it becomes very easy. You presented in a very simple way, and definitely I believe that our participants would be very benefited with this lecture. I want to congratulate to you and your team for this research work you have carried out, and many of us, we can understand that how much it is difficult to manage this kind of research, to carry out this kind of research along with the administrative responsibilities you have already. So this is it reflects your multi-task management skill. So thank you very much, sir, again. And now if anyone want to ask something to Professor Gaurav, then he may ask. If anyone,
if any query related to our work, I will be happy to interact with you all.
So if anyone wants, then he can open his mic and ask directly to Dr. Gaurav. Okay. If you have any query, you can write to our WhatsApp group, and we will extend it to the Professor Gaurav. Now I request to Yashpal Singh ji to please extend a vote of thanks from all of us on the behalf of the organizing committee and School of Agriculture Sciences, IFTMU University.
Thank you, sir. Good morning, everyone. Dr. S.S. Gaurav, Professor and ex-HOD, Department of Genetics and Plant Breeding, and Dean Faculty of Agriculture, Chaudhary Charan Singh, University Campus, Meerut. I would like to extend my deep sense of gratitude to you, sir. It is your fortune that we have got such a learned personality with us. Thank you so much, sir. I'm also I'm also thankful to K official of University Administration, Director School of Agriculture Sciences and Engineering, Professor Virender Singh, and participate of Faculty Development Program. I present my sincere thanks to our University Administration for organize of 1 week e-faculty development program on inclusion of innovative research in education for smart agriculture. I'm also thankful to organizing secretary, convener, and person who are directly and indirectly involved in this faculty development program. I wish you all good luck. Thank you, sir. Over to Dr. Himanshu.
Thank you. Thank you. Thank you to all.
Thank you very much, sir. Thank you, Gaurav, sir, for accepting our invitation and giving your precious time to us.
I'm also very happy to attach with your very important program. So I also thank and convey my all regards to all committee members, especially Himanshu, Virenderji, Jaspalji, because I know you personally, that's why I'm putting your name sincerely. So for thanks for your inviting me and organizing such a wonderful program. Thank you.
Thank you, sir. We are honored, sir. Thank you. Thank you.
Thank you so much.
May I leave now?
Yes, sir. Thank you. Information for all our participants that our next technical session 3, it will be start at 4 in the evening, and a scientist, a senior principal scientist from NBRI Lucknow will be speaking on the development, characterization, and documentation of new varieties of ornamental through an innovative way. So I request you to be on time. We'll try to start it on time as this session was. So be on time. Dr. R.K. Roy, ex-senior principal scientist, and he was the head of the botanical garden section of NBRI. He will be speaking on the topic. So please be on time. Thank you very much. Thank you all the participants, guests, and our member of the organizing committee, chairman, co-chairman. Thank you all. Thank you very much.